This method can help answer key questions in coral cell biology, such as what types of tissues and cells express specific types of genes. The main advantage of this technique is that you have visual information on RNA expression. Though this method can provide insight into spatial gene expression in adult corals, it can also be used in other life stages of corals, as well as in other invertebrate systems. Generally, individuals new to this method will struggle because there's so many steps and solutions to keep track of. So they need to pay attention to where they are in the protocol and keep good track of time when doing the washes. Kevin Rodriguez, a technician in my laboratory, will be demonstrating this procedure.
To begin, de-wax thin-sectioned paraffin-embedded slides with 100% xylene in glass Copland jars for 10 minutes under the hood. Next, fill 4 sterile glass Copland jars with 100% ethanol, 80% ethanol, 70% ethanol, and 60% ethanol, respectively. First, transfer the slides to the Copland jar containing 100% ethanol and allow them to soak for 10 minutes. Then, transfer the slides to another Copland jar containing 100% ethanol. Repeat this process with each of the Copland jars in decreasing order of ethanol concentration. First, transfer the slides to a sterile slide mailer with 18 milliliters of PBS. Then wash the slides for 5 minutes at room temperature.